Imagine a library that holds the biological secrets of millions of people, collected over decades. Now, what if every single book in that library had the same tiny, misleading typo, making it almost impossible to read? Well, today, we're diving into the technology that's finally fixing those typos. For years and years, scientists have been building this incredible library of human biology, carefully storing tiny pieces of tissue for future research. But here's the catch. The very chemical used to preserve these precious samples has a hidden flaw. It's been silently corrupting the genetic code inside. This is the story of how we're finally cleaning it up. So here's what we're going to cover. We'll look at the huge problem researchers are facing, the tiny chemical error that's causing all the trouble, the really innovative solution that's been developed, the hard data that proves it works, and finally, why this is such a game changer for medical research. So what exactly have scientists been up against? We're talking about a literal treasure trove of biological data that, until now, has been incredibly frustrating to read accurately. Okay, so the standard way to preserve tissue is called formalin-fixed paraffin-embedded, or just FFPE for short. It's allowed us to create these vast biobanks of samples that are absolutely crucial for studying diseases like cancer. But, and this is a big but, the formalin, which is the chemical that preserves everything, actually damages the DNA. It creates these tiny errors or artifacts that can look exactly like real genetic mutations. And that really is the heart of the problem. Imagine trying to read some critical instructions, but a specific letter keeps getting swapped for another one. In genetics, a mistake like that isn't just a simple typo. Oh no, it can completely change the meaning of the genetic code you're trying to figure out. All right, let's get into the nitty-gritty chemistry of this typo and see why it's so dangerous, especially in the high-stakes world of cancer research. It all boils down to a chemical reaction called cytosine deamination. The formalin preservative causes one of the 4 letters of DNA, cytosine, the C, to change into a different molecule called uracil. It's a really subtle change, but one with some huge consequences. This slide really breaks down how that one little chemical change creates a whole cascade of confusion. So in healthy DNA, C is always supposed to pair up with G. But when formaldehyde turns that C into a uracil, our modern gene sequencers get totally confused. They read the uracil as a thymine, or T, and just like that, a C in the original DNA has been mistakenly read as a T. And this is why it's so incredibly critical. This C to T transition happens to be a common type of real genetic mutation found in a lot of cancers. So this chemical artifact can look identical to a real sign of disease. This leads to false positives, and that can send researchers down completely the wrong path, wasting precious time and money. With so much at stake, this problem was just screaming for a solution. And that's where QIAGEN's new technology comes in, designed specifically to go in and fix this artifact problem. The Gene Read Kit is really engineered to do 2 crucial things. First, it extracts the absolute maximum amount of DNA from each one of these precious samples. And second, and this is the key, it uses a special enzyme to actively find and repair those chemical typos, those uracil bases, before the DNA is ever sequenced. The whole workflow is pretty streamlined for efficiency. After you get rid of the paraffin wax and break open the cells, you get to the magic step, enzymatic digestion. This is where the enzyme specifically targets and removes those uracil artifacts. After that, it's a pretty standard process of purifying the now clean DNA. Of course, a new scientific tool is only as good as its results. So, does it actually work? Let's look at the data and see the proof for ourselves. And wow, this right here really illustrates the before and after. Because the DNA damage from formaldehyde is random, the artifacts show up at a low frequency. This chart shows that the Gene Read Kit removes over 90% of these low-frequency C to T transitions, the ones most likely to be these false artifactual typos. But here's the really clever part. A true mutation that's present in a lot of tumor cells is going to appear at a high frequency. This chart shows that the kit specifically leaves these high-frequency mutations alone. It can actually tell the difference between random chemical noise and a real widespread biological signal. And this table just drives the point home. This is data from a real 15-year-old liver cancer sample. Look at the top 4 genes, MSH6, FGFR3, HNF1A, and CRE-BP. With the standard kit, they look like low-frequency mutations. But with the Gene Read Kit, their frequency drops to 0. Poof! They were just artifacts. And meanwhile, the true mutations, like an ALK and EGFR, are totally preserved. And it's not just about cleaning the data. The kit also excels at getting really high yields of usable DNA. When you're working with tiny, irreplaceable samples, every strand counts. And more DNA simply means more opportunities for discovery. So, what does this all add up to? Let's zoom out and consider what a tool like this means for the future of genetic research. The takeaway is simple but powerful. By systematically removing these chemical artifacts, scientists can finally trust the data from these massive FFPE archives more than ever before. It eliminates the noise, so they can focus on the real biological signals, making genomic sequencing a much more reliable and powerful tool in the fight against diseases like cancer. You know, the source material really sums it up perfectly. This technology directly addresses a major bottleneck in research, dramatically increasing the accuracy of studies that rely on this massive library of stored human tissue. Ultimately, this technology doesn't just improve a lab process. It reopens a massive library of biological history that was previously so hard to read. It's like finding a clear translation for millions of aging, invaluable books. And the question it leaves us with is, what have we been missing? And what new